tired, probably.
Not bad.
Good. Glad to hear that, Carlos. Great. I was just responding to Carlos. He said he's fine. They're chatting on the window.
Oh, yeah. I'm ready for Friday afternoon. I'm sure some people are past their Friday afternoon, I think, by now, right?
That's right. That's right. Well, we'll get started in a couple of minutes. Let additional people join. It's always with the virtual workshops. You start off with many people on the first day, and it sort of whittles down towards the end of the week. Were you able to resolve with you and Lois the Galaxy issues, or?
No, actually, she has a G. It's about data upload. So she has a G drive that has her data on it, and we use the Globus data transfer, and it failed. But her endpoint name has a space in it. I wonder if we were wondering if that's a problem.
Okay.
I kind of think it might be, because that could be interpreted as the parentheses and stuff like that.
Interesting. Okay. Well, we should give it a try. You said the data is in a Google Drive somewhere?
Oh, it's in her. No, it's in a hard drive.
Oh, an external drive. Yeah. I know that works because I helped somebody do that a few months ago, and they had it on an external drive.
Yeah.
Yes.
It depends on your system, but sometimes you have to explicitly tell the Globus endpoint that it's okay to access your external drive.
That's right.
I
Yeah.
can't
good
remember
point.
exactly how you do that though.
You have to give the endpoint authorization to access it. You're right. Yeah, that's a good point. So we should look into that.
Okay. Yeah. Well, here's the error message we got. File not found, scratch, buried.
So that looks like a potentially different error message to me. There have been some issues over the last couple of days with Galaxy mounting file systems, and that
Right.
looks like it could be related to that. So in that case, we'll just have to wait for Galaxy to resolve it, I think.
Okay.
Okay. Yeah. So for Lois, we've had some issues over the past days. We have some conversations going with the people who manage it, and Kathryn says this might be part of that issue instead of just us not accessing your endpoint well.
But it is also worth checking that your endpoint does have access to your hard drive as well, because that's a quick fix if that does turn out to be the problem. So
hopefully
Lois, it might be worth just making sure that one of the instructors has your email address so that we can.
We do, yeah.
Yeah, we can just contact you when once we know that Galaxy is working in and check that everything's all right.
I just put it in the chat box. It's not private by any means. Anybody can email. And I appreciate the help. I didn't realize when I was creating the endpoint name that what I was doing, and I wouldn't have left a space in it if I knew that would be some fatal mistake.
Okay, I think we probably should get started. We have a good number of people here. Start by Sharing my screen. Okay, so welcome everybody. Last day of the workshop. I know it's hard with virtual workshops. It's obviously very different from in-person workshops. Just mainly the amount of time we have to spend with each other, and obviously giving people individual attention is much harder in the virtual world. But hopefully, people learn some things this week, and today we'll cover some more aspects of the resource that you will hopefully find useful. So what we're doing today for the last day is we'll have a couple of lectures to start off with. One, we're going to focus on, again, on Galaxy output. Specifically, we're going to be looking at the output files from an RNA sequence analysis. And we will, but before that, I'll give a short sort of general lecture about RNA sequence analysis and what steps one would have to take to analyze an RNA sequence data set. It'll be a whirlwind. We'll try and go through these very quickly, just again, because of time. We do have various recordings, as hopefully you've learned so far. We have a YouTube channel full of recordings. And since we do multiple of these workshops in different flavors, you will find some of these lectures that are more extensive. And so I would highly recommend, if you're interested in doing analysis in Galaxy, or specifically RNA sequence or variant calling, we do have some recorded lectures that are a bit more extensive and may give you some more information. And of course, always email us and let us know if you have specific questions. We can point you to specific YouTubes or answer them. So let's get started here. Okay, so I'm going to go through a bunch of slides. I adapted these from slides from Kathryn. So I'm going to tag team with Kathryn. I'll present these slides quickly, and then Kathryn is going to walk us through some of the output files. I'll keep sharing my screen since I have a bunch of output files that she can describe. All right, so this is again just a very general overview. And again, for some of you, this may be old news, and for others, maybe it's new and interesting stuff. So the first question when you're thinking about doing a transcriptomic experiment is why do you want to do it? And there are multiple possibilities. So you can, one is that you can capture a full unbiased repertoire of transcripts for gene models. So for example, you are since you are not really using, for example, a microarray, which already has a predetermined lists of probes on it, which may not be comprehensive of the whole genome, depending on the genome annotation status. The transcriptomic experiment is when you do the experiment, it's really agnostic about the genome. And in fact, if you're doing an experiment that has a host and a pathogen, it's agnostic of that as well, right? Unless you're doing something specific to enrich for one or the other. You'll be sequencing whatever is RNA that's captured in your assay. RNA sequence data sets allow you to do ask some functional questions, like what genes are up or down depending on the condition, stress or drug treatment, or different isolates. And then, of course, as we learned yesterday from looking at RNA sequence data in the genome browser, you can use RNA sequence data to explore gene models and splice junctions and determine potentially alternative splice variants of genes. So again, just to think about this some more from a biological standpoint, I did this a little bit a couple of days ago when I was drawing things out. But basically, in your sample, you have your DNA, which is making your pre-RNA, and then making your transcript, your mature RNA. This mature RNA is what you take into your analysis in the lab, where you fragment the RNA, you make a cDNA, and depending on the RNA-Seq type of RNA-Seq you're doing, you may have different modifications or use different kits. But ultimately, you end up with fragments of cDNA, which you sequence, typically with something like Illumina sequencing. And that will generate then sequence reads, and that's the Fastqc file, which we showed briefly yesterday, which you take out now and you everything becomes in silico. So you start with an in vitro setting, in vivo setting, you go in vitro with your preparing your samples and sequencing, and then in silico is the actual analysis. So you're starting with a file that's full of reads, and then you have to map it back to the genome, and then you can use various analyses to determine gene models, see where the reads are aligning, determine expression values for genes, and so forth. So there are many considerations to take when doing an RNA sequence experiment. I'll go through a bunch of these quickly, and there are obviously more of these that you can consider depending on the experiment you are doing. Do you have enough biological replicates? So the recommendation is always having 3. Some people say, well, having more is better, but you'll reach a point where more replicates for general RNA sequence experiment is not really going to make a huge difference. So the recommendation is to have 3 biological replicates. Having biological replicates is super useful because there is variability in RNA sequence in expression and also capture of the RNA in your analysis. And so so it's always good to have more than one at least, especially if there's a set of genes that you're really interested in. You want to make sure that they're well robustly represented in your analysis. Do you have enough RNA? So this, of course, depending on what you're trying to capture, if it's something that is rare or you have a low amount of your sample, then of course you might not capture rare transcripts in your sample, and so that's a consideration to keep in mind. And you may have to sequence more deeply to pull those out. Again, if you're sequencing, you have to consider your sample and does it contain the RNA that you're interested in, and so that's another consideration, of course. Are you using the appropriate selection method? Again, that's where it's important to think about how how you're going to process the samples, and of course how you process the samples may affect the results. We were talking about long read RNA-seq the other day, and I always forget that some of the methods are really 3' biased, and of course that then will affect how you interpret the results, and so it's super important to do that. I know Edith is indicating in the chat that also having enough RNA is important, and the quality is also very important. Absolutely. And so that's absolutely important. If your quality of your RNA is degraded, then of course that's going to affect the abundance of your RNA, and you will not see your what you're interested in, or you may get a biased output from your experiment. Are you interested in doing a stranded experiment? And again, most kits, from my understanding right now, are basically stranded kits. So often these days, and I know this from the work we do in aligning reads data from people's experiments into our resources, is that often even the experimenter is not really aware that their sample is strand specific, because that's really not what they were interested in. They were just trying to get expression values for their genes, and having it, knowing, doing a strand specific library in that case maybe wasn't their primary objective, but it's possible that their results are truly strand specific. And so that's something you have to contend with. But of course, we talked yesterday about how the strandedness of your results could be quite useful for determining, obviously, especially if you have genes that are very compact, or a genome that's very compact, and you have potentially overlapping genes, and getting the stranded RNA is super important. And then of course, the last thing was, do you have appropriate controls? I don't have anything under this, but I think that's an obvious one. Always think before you set up your experiment, think about what controls you need. And this really relates back to really what is the question that you're trying to answer with the experiment. And I know some people who manage core facilities where they work with different research groups to prepare their samples and sequence them. they often have a meeting, if you have a really good core facility, and they will meet with you before you even start the experiment, or at least they have that service available. And I would highly recommend that you take advantage of it, to sit down with them, and sometimes one of the first questions they ask you is, well, what is the purpose of your experiment? What are you trying to do? And again, we talked about this, I think, so one other day. It's always, everybody wants to do RNA sequence experiments or whatever experiment is out there, and sometimes you forget that, well, is this really the most appropriate method to use? And if it is, then to think very carefully about the controls that you need, so that you can actually, in the end, when you have millions and millions of reads, that you don't all of a sudden have millions of reads that you can't really interpret. And so that's super important. Okay, so what can we learn from RNA-Seq data? I think that was, we talked about this a lot, but I think this is just highlighting, this is a view from a genome browser view. We're looking at, I think this is a fusarium gene, which has exons and then introns right here. And then in the lower part here is a coverage plots. This is a bigwig file, basically. And you can see where there's a peak, that's where there was RNA mapping back to the genome. And then where there's a trough here, that's where there's no RNA-Seq RNA reads mapping to the genome, which would typically align to the introns, or of course, it could align to intergenic regions. And so having the RNA-Seq data will help you determine the gene structure, and also will help you determine the UTR regions. And so often, many of the genomes that are annotated, they don't have any UTRs annotated, because they're actually quite difficult to annotate computationally. It's hard to know where transcription starts or ends. There are some features you can look for, but it's not exact. And having RNA sequence data to support that is quite useful. Okay, and of course, having different samples that potentially show different intron splice junctions, splicing could be useful for determining alternative splicing, and potentially alternative splicing that's connected to a developmental stage of your particular organism. Okay, and so you can use RNA sequence data in addition to looking at splice junctions and intergenic regions. You can use the RNA sequence data to determine differential expression between genes. So for example, this gene here on the right, you can just eyeballing it, you see that it has a lot of coverage. And then the gene here, the gene, sorry, on the left has a lot of coverage, and then the gene on the right has low coverage. And so obviously, eyeballing it doesn't necessarily tell you that these are truly differentially expressed, but it gives you an idea that this gene has a lot of transcript and this gene has low transcript. So having biological replicates and being able to do a true differential expression analysis on this would be quite useful, because that's where normalizing your expression values comes in, and it's very important. Okay, so how do we get to a point where you have reads that you can look at in the genome browser, and reads that you can use to determine the expression of each gene, and then use that for differential expression? So this is just a schematic showing you starting up with your sequencing data, your Fastqc files, and you can do a couple of things with this. I'm sure you can do other things, but for our purposes, we'll talk about a couple of things. One is you can take these and de novo assemble RNA transcripts, and you could, and there are different tools for doing that. Trinity is one of them. And then once you have your transcript, so this would be assembling these de novo transcripts would be agnostic of your genome, right? So that's, and you can see this often with there are different situations where you do this, but an example would be if your genome is not sequenced. You're working with an organism or a genome that's poorly annotated, and you want to assemble transcripts straight from the RNA data. And then you can take that information from these assembled transcripts and do different things with it, including differential expression, or look at what genes are there, do some functional enrichment, and so forth. most typically people do is they take the RNA sequence data and they map it back to the genome. And that's basically, as we were showing in the previous slide, go back here. So mapping it back to the genome will give you something like this. So imagine instead of a graph here, you have reads aligned here. And typically when you're mapping this back to the genome, again, there are different tools. One of the more standard ones is HiSat2, STAR is another one. In our workflows, we use HiSat2, and we'll show that briefly. But that's going to try and align your reads to the genome. After mapping, you can also visualize the data in the genome browser. You can use this data to predict gene models. You can use the mapped reads to determine differential expression. And then, of course, once you have differential expression again, you can do some functional enrichment and look for genes that are functionally enriched or genes that have a particular Go term, for example, or any other function that you can assign to them and see if they're enriched in your different samples, differentially enriched in your different samples. Okay, so there are multiple steps that go into an RNA sequence analysis, and this is, pretty much what you can do in Galaxy using our workflows, and also typically what one would do when you do your own RNA sequence data. So going back here, the first step is the quality control. And actually, before that, just showing you, I showed this yesterday briefly, but this is just a little synapse of a Fastqc file. And as I mentioned before, a Fastqc file is just a text file, just like a FASTA file. It just has structure. And the structure of a Fastqc file typically starts with an @ symbol. There's information here about the sequencer or the location on the slide, and potentially other information in here. This becomes useful. In most cases, people ignore this line. You're not going to look at this, but there will be cases you may want to go back and look at this, and that's if you're doing multiple experiments over a long period of time, and you're trying to compare things to each other, and this could help you a lot in determining any batch effects. Were these done in the same sequencer? Were they done? Was there potentially a different location that ran your sequencing, and those could actually affect your results and may end up being important for you to go back and review these, especially if you're taking data from the sequence read archive from, let's say, one large study that spanned over multiple years, for example. You'll often find that the sequencing was done in different locations by different people, and there are some well-known studies that went back and looked at some of these large studies and looked at the location or the sequencer that was used for the sequencing, and they found that there's actually a batch effect in the results. That the results that they were showing in the paper was really more correlated with where the sequencing was done as opposed to location of expression, like liver or brain and so forth. And there's some very well-known studies that have been affected by this batch effect. The second line here is the sequence. So this should be very familiar to everybody. There's A's and T's, G's and C's. You follow by a star, a plus symbol, and then followed by the quality score. And again, I won't go through this in detail. I mentioned it briefly yesterday, but the quality scores shown here on the right in this table. But the reason why ASCII code is used instead of, because one would say, well, why don't you just put the numbers in here instead of these letters, and save us the calculation of converting this to numbers. And the issue here is that you're able to save space by using a single character instead of 2-digit characters. And this ends up being an issue when you're dealing with millions and millions of reads and multiple samples. And so it seems like a trivial thing, but it actually has a good purpose. Obviously, you're never going to go in and look at this manually. And and try and determine manually the quality score for each of these reads. That's done computationally. There are too many reads for you to do it manually, and that's where the quality control step comes in. And typically, people run something called Fastqc, and Fastqc is a tool that will take a look at all of your reads and give you some output about the quality of those reads overall over the entire sample. And we will, again, I won't go through this in detail here because Kathryn is going to walk us through one of these examples and show us some of the quality output and how you would interpret that data. The next step after quality control, there's a trimming step. So you trim so based on the quality control, you may, the trimming tool may take out some of the reads that are poor quality, and also take out any of the adapters or linkers that were used in the experiment that you want to get rid of, so they're not affecting your alignment. Read alignment, and so this is, again, imagine doing a blast, but you're doing a blast now on millions and millions of reads, and so that would take forever, obviously, to try and align your reads back to the genome. And so the methods that are used are really is just trying to find the best alignment that you can get from a particular read aligning to the genome. And the idea here is that this is an example here just showing you two sequences, and you're trying to align them to each other. So if you try and imagine how to align this, you can think of a couple of ways how to align this. And typically the heuristic that's used to determine which is the best alignment is a pretty straightforward one. Basically, if you have a match, it gives it a score of 1. If there's a mismatch in the alignment, it gives it a -1. If there's a gap, it's a -2. And it basically adds them up and determines very quickly which one of these is the best alignment. It may not be the perfect one, but it works really well. You can imagine aligning these in many different ways. You just want to pick which one is the best alignment. And so the aligners will do this and do this for you, obviously, but they're using a pretty straightforward method to calculate which is the best alignment. Okay, so remember when you do your read alignment, you're starting off with RNA that came from spliced messenger RNA. And so again, I described this the other day, but your reads are coming from all over the place. They're coming from the exons, they're coming from the UTRs, and they're coming from the junctions where the these are 2 exons, for example, they're coming from where the 2 exons were spliced together. And so these will be these reads here. And so when you map them back to the reference genome where your gene is annotated, of course, that in the genome, the gene will have, the exons will be interspersed with introns. And so when you map these back, your reads that align to the red exon will align to the red portion of the genome where the red exon comes from. The blue ones will align to the blue one, and then your intron spanning reads will be split. And that's what we were looking at in the genome browser to look at evidence for splicing. And so anytime you get a read that's split, that means there's an intron here. And the more reads you get, obviously, for a particular location, the more confidence you have in that this is an actual intron. Okay, so the mapping tools, they need to be capable of aligning millions of reads, and it needs to be done in a reasonable amount of time, right? You don't want to do this and then have to wait weeks before your alignment is done. So this needs to be doable in a quick way. The algorithm used needs to be good enough. You're not looking for perfect alignments, and this turns out to be actually really good, believe it or not. Depending, on what you're doing, but in cases, especially if your genome has introns, then you want to make sure that your aligner is capable of detecting these intron spanning reads. So it needs to be a splice-aware aligner. The couple of ones here I'm mentioning briefly. HiSat2, it's fast, but it's a little bit less sensitive. STAR is a very sensitive tool, but it's slower. And so again, it depends on what you're doing. So once you have your alignment, right, you have to then, in order for you to say something about the expression of each of these genes, you have to somehow convert these reads, the aligned reads, into some kind of a number, a count. And so that's where you use a tool that will determine the count numbers per gene of interest, or per entity of interest that you map to, basically. Okay, so okay, and there are some considerations to take keep in mind, and so you can imagine that there are genes that are overlapping, and so what do you do with reads that are in the region of the overlapping section, right? And so, of course, if you have a strand-specific experiment, if you did a strand-specific library, then you can use the strandedness of the RNA to determine which gene the reads are coming from. You can also have situations where a read is aligning to two genes, right? So what do you do with that in that case? This is considered an ununique aligner, right? And there's some methods, they average the reads that map to more than one sequence. Other ones, they basically take these reads and assign them to one of the genes. They basically say, well, we don't care, we're just going to assign all the reads to one of the genes. And other ones just throw them out. You can set those parameters in many of the aligners to determine to determine the expression value of the gene. In our case, in VEuPathDB, we don't use them to determine the expression value of the genes. We essentially throw them out. But what we do is we show you in the genome browser. You may have noticed that some of the reads are called non-unique aligners. And so what we do is we allow you to look at those, because sometimes it's useful to see if you have a gene that seems to be low expressed, or it has a region that has low expression for some reason. Turning on the non-unique aligned reads will help you then determine, oh yeah, this region looks like it's coming from a highly repeated region in the genome, and then you will see peaks there in the non-unique aligned coverage plots. Okay. So once you determine a count, right? So again, these are some parameters here that again, for the sake of time, I'm not going to go through them in too much detail. But what's super important though is to sort of make some decisions as to what are you counting. So one example here is to think about whether you're going to count the uniquely aligned reads to one feature, or are you're gonna remove them and so forth. Another thing you need to think about is to determine whether you're counting the reads per exon. For example, if you're interested in looking at alternative splicing, you may want to count the reads per exon, because you want to then determine if there's a difference in exon expression per gene in the different samples. Or you can obviously count the reads per the entire gene, right? And so, the entire transcript, basically, if you're looking just at differential expression. And there's a lot more information available that you can read about these and go through them. Okay, so differential expression is used obviously then to compare different samples to each other to determine if there's a difference in the abundance of the transcripts between different conditions. And these could be between a knockout and a wild type, different life cycles, for example, different nutrition sources, different virulent states, so you can take then the coverage plots, obviously, which we already saw from the previous images that I showed, that you can see visually, you can see that there is a difference between genes in the coverage, and then you can count those. But the question is that number, the value you're getting for the expression, is that real or not? And for that, you have to use a tool that's going to normalize your read counts and give you values that are statistically robust. And that's where obviously having biological replicates is important. Okay, and so this is an example from I think this is an experiment that Kathryn did, but this is an example of these are different samples, and you have the counts here on the y-axis, and this is an unnormalized situation, and so and each one of these is a different sample, and you can imagine a situation here where the gene in this green X is expressed in both of the samples, but because these are not normalized, then these two genes look like they are not differentially expressed. So after normalization, though, after you normalize the counts, what your samples will look like, they will look like this. So you've centered the midpoint, and now what you can see is that these two green X's are actually potentially quite differentially expressed in between these two samples. And so that's where normalization is super important, because in this case, you would have probably just looked at the counts and said, well, the counts are very similar. It doesn't look like this gene is differentially expressed, but that's before normalization. And so that's where the normalization becomes very important. Okay, so let's see what I want to say about this slide here. So of course, depending on the experiment you're doing, you may have multiple conditions. And so typically, so the more samples you have and the more conditions you want to integrate in your differential expression, obviously it becomes more complicated. Typically in Galaxy, you can run multi-factor experiments and analyze them. It gets a bit harder, and the more sample you have, it gets more and more difficult. So at some point, you end up having to work with the bioinformatician. But being able to run a single factor experiment in Galaxy, so comparing two conditions, it works quite well and super easy to set up. So it's a nice way for you to do. Let's say you have an experiment with three conditions. Really what you like to do is put everything together and do a multi-factor analysis. So you can do differential expression between all the samples. But as a single factor experiment, you'd compare, for example, male samples to female samples, and then maybe, or let's do a different condition. You can compare knockout 1 to knockout 2, and then knockout 1 to the wild type, and then knockout 2 to the wild type, right? In a single factor experiment. In a multi-factor analysis, you would compare them all together in one differential expression. So the most commonly used tools for determining differential expression are DE-Seq2 and EdgeR, and I'll let Kathryn maybe mention a bit more about these when we're looking at the data. That we'll look at some DE-Seq data output, but typically they're going to run some kind of a statistic on your reads and then give you some values that are typically log twofold values, showing you the difference in expression between the factors in your experiment. And they also provide you with additional statistics. Most typically, you'll get a p-value, but also a q, what's often called a q-value, or an adjusted p-value, or FDR adjusted p-value. And that's a p-value that's very important to look at when you are analyzing an experiment with multiple conditions, with many conditions. I think I do have a slide for this later on. The only thing I'm going to mention here is that if you can imagine with a test that has 10,000, well, if you have a test that has 100 genes and you're using a p-value of 0.01, then you would expect that 1 in 100 of your genes is by chance is false, right? Is a false positive. But if you have an experiment that has 10,000 genes or 20,000 genes, then a p-value of 0.1 will give you potentially 100 false positives. And so the more the tests you're running basically in your statistical analysis, it becomes more likely that your p-value is giving you a value that is that starts giving you too many false positives in your results, right? You'll start making conclusions about the results that you have, and what will happen is, because you have so many tests, that you get just a higher than a comfortable number of false positives in your results. And that's where the adjusted p-value comes in very handy, because it will adjust for these many tests, and then you can be a bit more confident about the false positive rate in your results. And typically the Q value is going to be lower than your p-value, and we'll see this in one of the files. You'll see that the adjusted p-value is always lower than the p-value. And so I highly recommend always look at the adjusted p-value when you're doing an experiment with multiple tests. This doesn't only apply to RNA-Seq experiments. We'll talk later about Go enrichment, and the same thing applies. I think I have pretty much the same or a very similar slide in that lecture, where we have an adjusted p-value, and always look at the adjusted p-value because of to account for the multiple tests that you're running. Okay, so this is just, there are different packages that you can use for doing differential expression. We use DE-Seq2. It's they all have different advantages and disadvantages, but they're all pretty robust and heavily used. And there may be situations where we're talking to a bioinformatician about what is the best method to use for differential expression might come in handy, because people with experience with these will be able to give you some ideas about where one of these works a little bit better than the other. But in most cases, they're pretty robust. Okay, and I think this is my final slide. This is showing you a workflow in Galaxy. So remember yesterday I built a very small, simple workflow of a multiple sequence alignment. So this is a little bit of a more complex workflow. This is a workflow that's analyzing reads from two conditions. And so you have input sequence here at the top, and you have another input sequence here from sample 1 and sample 2. And the input sequence that you're putting in, so you're feeding the workflow your Fastqc files, that's going to go in and going to get processed in different ways. So each of these workflows are basically identical to each other. You're basically doing the same thing on each of the samples independently. And these go through the first step here is this Fastqc step. And the Fastqc, that's going to give you the quality, give you an output about the quality, and we'll look at that in a second. Fastqc, some of these steps, it's not super important to know exactly what to do, but the Fastqc groomer is just going to prepare your files, make sure they're in the right format. You could actually skip this Fastqc groomer step. You can still run your analysis without doing it. This is just to ensure that your files are in the right format for the next step. Trim-o-matic is a tool that's going to trim your reads. So there are multiple different types of tools that trim reads, including Trim-o-matic, Sickle, and many other ones. So this one will go in and trim out all the standard adapters and also low quality reads, and you can modify the parameters too if you want. HiSat2 is a tool that's going to align your reads. Remember, so we after you do your quality control and trimming, you're going to take your reads and align them back to the genome, and so that's a tool that's going to generate a file that basically has all your reads in it and the locations where they're aligned to in your reference genome. Typically, if you want to visualize these, you can visualize the output of a HiSat2 analysis, and that's typically a file called BAM file. You may have heard of BAM files. And a BAM file is a binary file. If you try and open it in a text editor, you can't read it because it's a binary file, and it's huge. It contains all the information about the reads, where they align, and the reads themselves. So if you took a BAM file and aligned it, you can upload it to the genome browser and align it. You will get coverage plots. And if you zoom in all the way down to nucleotide level, you'll see all the reads and where they align. And you'll be able to look at the actual nucleotides for each of the reads. As a consequence, these files are big. So remember, you're starting off with millions of reads here. So files that are gigabytes, potentially megabytes or gigabytes in size. And as you go through this, your data files are going to expand. So you're going to start with maybe a gigabyte here, and you may end up with 5 gigabytes here after you align and generate the BAM files. One way to quickly and a little bit easier visualize the data is you can convert this BAM file into something called a BigWig file. Again, there are all these different names, but what you're doing, what the BigWig format is basically taking a BAM file and stripping out all the information about the reads themselves. It's just simply taking the information about where the reads align. So it generates a histogram basically of the aligned reads, but removes all the information about the reads themselves. So you can't zoom in and look at the nucleotide level. All you're going to get is the coverage plot. It's in most cases, this is all you need. You don't need to go down to the nucleotide level. And so BigWig files are quite useful. They're smaller. So as a consequence, anything you do with this will be faster than working with the BAM files. Remember, now you have these reads aligned, so you need to somehow generate counts per gene, and we use a tool called HTC count, and this generates for each gene in your genome, it will generate a value that's basically based on essentially counting the reads that align to each of the entities that you're looking at. And this is where you can actually set parameters if you want to only look at exons, if you only want to look at the transcriptional unit. So you can really determine what to look at. All of these alignment and HT counting are tied to your reference genome that you're aligning things to. And so, often you have to, if you're not aware of what the reference files look like, you may get some errors here because HT-seq has some default parameters. And if it's, for example, if HT-seq is looking for an entity called gene in your file, and for some reason your file genes are not called genes, they're called something else for whatever reason, it's just not going to find them, right? It's looking for the word gene, and if it doesn't find it, it's going to say, well, I can't find any genes in your file, right? So that's where you have to kind of you may get errors sometimes that are simply based on making sure that you have set the parameters for HTC count to fit with the format of your reference genome files. Okay, once you have your counts now both of the counts from your this is a two-factor experiment, a one-factor experiment, 2 samples. What you do then is you merge them together into a tool called VEuPathDB. So this is where it's going to compare the expression values for both of these and run the statistics on them to determine if there's true differential expression. And remember, these are coming from these samples that you're putting in here are typically, if you're doing differential expression, they actually have multiple replicates, right? So each one of these is not going to be a single RNA-seq experiment. It's going to be, wild type replicate 1, wild type replicate 2, wild type replicate 3. And so that's going to go all the way through here. And that's going to be super important for VEuPathDB, obviously, to determine the differential expression between the genes. Okay, so Lois is asking, where does the normalization take place in this workflow? So there are really, I guess, 2 places where normalization, you can imagine normalization taking place. So HTC count is I believe HTC count does some kind of normalization. Kathryn does not.
No.
Okay, no normalization.
No,
so the only normalization
just counts
is here,
the
right?
reads. Yeah, it's just in
Okay.
VEuPathDB.
So just VEuPathDB is where the normalization is happening. Yeah. Oh, that's right, because HTC count just generates transcripts per million, right? So.
No, it doesn't need, that's normalization as well.
Oh, that's normalization.
Does,
that's right. Yeah.
All HTC count does is it counts the number of reads aligning to each gene. That's it.
That's right.
Raw
yeah.
counts, no normalization there at all.
And if you convert these to transcripts per million, that's right. The transcript per million is a normalization, and that's something you can do. So when you look in if you're doing a full change analysis in VEuPathDB resources, and you're comparing samples to each other, those are coming from their TPMs, and so they're normalized within the sample, not between the samples, obviously. And so within
we do have another tool in Galaxy that's not in this particular workflow that takes the HT-seq count input and makes TPM values as well. So you can use the DE-seq2 normalized counts, and that does the normalization in a slightly different way, or you can run this other tool, which I think is called HT-seq count TPM or something. It's something fairly obvious like that, and that will help you make the TPM values if you want to.
And in fact, when you transfer TPM values, I showed you yesterday how you can transfer data back to VEuPathDB using this export tool. The export tool, if you're starting with output from a workflow, the workflow I just showed you that does not generate TPM values, you'll have to first take your count files and convert them to TPM values in order to be able to transfer them. And you'll see here that we have a tool that will convert either to FPKMs, but typically that's what we use. So you can convert your HT-seq count output into TPMs, and then those are what you can transfer back to VEuPathDB. And we have a number of exercises that walk you through step by step to actually generate all of this here, and also generate the TPM values. And Suzanne, maybe later we can link some of the exercises we have for the Galaxy analysis, maybe the ones from the AGS workshop. Those would be quite useful to have them there.
Okay, I can do that.
Yeah, and Lois, you're right. The collection includes the triplicate observations. Yes, and so for example, if I'm looking down here at this workflow that I ran, it's comparing males to females. These were male female mosquitoes. And if I open one of these, you'll see that it contains male 1, male 2, male 3. These came from paired end experiments. So each one of them has forward and reverse reads. Okay. All right. So Lois is asking, maybe Kathryn, you can answer. Did you answer them there? Let's see. So
which question are you talking about?
I'm looking at both. So the question from Lois about the normalization. So she's asking, when the collections are compared in DE-seq2, the normalization occurs across the collections as well as within the collections?
Yes. So DE-seq2, if you remember that figure that Omar showed on one of his slides for the normalization, where it showed all the samples with the box plots. Maybe you could put that back up, Omar, actually, a second.
Yes. Mm-hmm.
Yeah, this one. What it's basically doing, and it depends on which tool, is it takes either the upper quartile of each sample, or in some cases it takes the geometric mean of each sample, and it just uses those to line all of these samples up, and it doesn't care when it does that. So yeah, you can see that there. I think this one's done probably an upper quartile normalization, so it's just lined everything up along here. Sorry, my line's not very straight there. Yeah, so when it does that, it doesn't care which condition a sample belongs to or what that sample is. Yeah, so this normalization was actually done with DE-seq2. In answer to your question about DE-seq is doing what was in the example slide, this is exactly what DE-seq2 was doing. So in this, the colors, we didn't really talk about what the colors were, but basically the colors were 3 different conditions in this case, and it's normalized just across all of them without actual reference to what those are.
Cool, thank you. And then a question from Carlos: in the log 2-fold change you mentioned, it is a measure of magnitude and direction. I mean, it's a measure of magnitude, but not necessarily of direction. It's just telling you something about.
No, it is a measure. It is a measure of direction, because if you once you convert it to log2, then if the gene isn't changing at all, the value will be 0.
Oh, no,
If
I meant
values-
direction. Sorry, in the question it was 3' to
yeah,
5',
sorry.
5' to 3', that kind of direction. You're right. Yeah, so up
Yeah.
or down for sure. Is that what you meant, Carlos? Maybe I misunderstood your question.
No, just to know what do you mean with direction? So direction in the sense of the strand or in another way?
No,
Yeah, no. So yeah, Kathryn was yeah. So what Kathryn was. So I thought you meant by direction, but it's not direction. It's the direction of expression, right? So whether it is up or down regulated or not regulated. Yeah.
Okay, thank you.
Yeah. And we'll look at the file right now, which will give you hopefully a better idea. Okay, so let's switch to that now, because I know, Kathryn, you have to leave in half an hour, so we'll make sure that we can get through this fairly quickly. All right, so here's an output. So this is a Galaxy workflow that I ran a couple months ago. This has output files, everything is green. One thing to note is that I'm only showing 8 output files. Let's call them files. They actually contain more than one file in them. But also you'll notice here there are 134 hidden files in here, which you can reveal. They're hidden just because there's a lot of output files that you won't necessarily care about looking at, but sometimes there are some files that you may want to go back to. So it's always good to know that these hidden files are there, and you can, by simply clicking on them, you can view them and look at them. I won't do it right now just because it'll make it super messy. So you'll see here that the females and males, these were the collections that I used. Each one has 3 biological replicates. They have, each one of them will have a Fastqc output, which you can take a look at. And in fact, this is the output on the collection. If I open this, you'll see that each replicate will have its own folder with Fastq output. If I click on one of these, you'll see that each pair will have its own output file. And if you look at that, you will get the actual web page that shows you the scores. And we'll come back to that in a second to look at what maybe we'll do a whirlwind view of what these different measures mean. The BAM to BigWig, this is the conversion of the BigWig file, the BAM file into BigWig files. And again, for each of the samples, it's going to create a single BigWig file. So the pairs are going to be in a single BigWig file for each of the biological replicates. But each one of these, I can take one of these and visualize it in the genome browser. And let's say in this case, it will be in VectorBase against the right genome. I can actually look at them. You can also export them to VEuPathDB and then view them through the dataset section. And then finally here, there's the DE-Seq2 results. Actually, I don't see the DE-Seq output. Let me show hidden files. You'll see these are all the hidden files, and you'll see that there are HTC count files in here, and DE-Seq2 is the PDF. So somewhere in here, there should be the actual table of DE-Seq results. Let me do a search. Oh, that's interesting. I'm missing that file for some reason. Okay, I'll try and find one. Maybe we'll when we get closer to looking at these. Let me go back here. Let's take another experiment. It doesn't really matter which one we look at. I'm going to switch to this one. Yeah, this one has results in it. I'm not sure what happened to the results in the other one. I must have maybe deleted it by mistake. But here's an output from DE-Seq2. It's a table. So when you click on this, it shows you the table. It has close to 14,000 lines, right? So each one of these is a gene. If we look at this, you'll see a table that contains gene IDs, And then I'll let Kathryn explain what these are in a couple of seconds, but you'll see here's the adjusted p-value, and then the log fold 2 is right here. And so for Carlos's purposes is you'll see that there's some positive numbers, negative numbers, and those would indicate up or down regulation, fold change at least. Okay, so let's just stay on this one and take a look at one of these Fastqc files. Okay, so Kathryn, you want to take it away here? Let me see if we can make this bigger.
Yeah, is there any way that you can hide the side panels?
Yeah.
Yeah, there we go,
here.
and then we can see the full width of it. So Fastqc is it's really meant as a quality control program, and the primary purpose of it is to read through your Fastqc files and get an idea of how good your sequencing was. So if you remember when Omar showed you the Fastqc files, it had the sequence, and then it also had that line which was all letters. And Omar was that a question? Omar was saying that those letters were.
Here, hold on. Let me find the person and mute them.
Mute them.
I think they muted. Okay, go ahead.
Okay, yeah. So Omar was saying that those letters in the Fastqc file relate to the quality. So what they are is the probability that each base was actually what the machine called it to be. So the primary thing that Fastqc wants to do is read your Fastqc file and read all of those quality lines. And as Omar said, you don't want to do that yourself because there can be 30 million or 60 million or 100 million of those lines in a file, and then you'd have to convert all those values back to numbers yourself. So Fastqc is going through and it's reading those, and probably the most important output from it is this. I'm pointing with my mouse now, but it's Omar's screen you're sharing, so that doesn't work. Is this plot at the top? So what we're looking at here, and this is actually not that good to see. The other one you had before was good to see, but I'm not going to make you go back.
I can, if you want, I can go back to it quickly if
it's
that's
more
better.
obvious. It's more obvious what's happening. Yeah. Back to the other one in the other workflow.
Yeah, I'll do that.
Yeah.
Let's go to the males and females, and can hide hidden back here.
Yeah, perfect. So you can see what's happening much better in this one. So on the, can you just go up so we can see the axes? On the x-axis of this, we have the position in the read. So this this particular sequencing run was generating reads that are around about 100 base pairs, and it's lined all of those reads up with the first base pair at the left of the left axis and the right the 100th base pair at the right of the x-axis. And then on the y-axis, what we have is the quality score after all those letters have been converted to numbers. So here we're looking at the quality score for the first base in every single read. And here we're looking at the quality score for the last base in every single read. And what we're seeing here is a box plot of how that's distributed across all of the however many million reads you have in your sample. And actually, if you just scroll up quite a second, you can see here how many reads that is. So this was, yeah, just shy of 20 million reads in this particular sample. So the reason it's easier to see on this one is because historically Illumina sequencing capped their qualities at 40, but newer sequencing newer Illumina machines don't actually do that. They allow the quality to go higher than 40, but Fastqc hasn't quite caught up yet. So the top of the y-axis here is always 40. So sometimes you'll see what we were looking at on the one we were looking at before, where all the box plots are actually off the top of the chart. And if you see that, that's fine. That means your data is good quality, so I wouldn't worry too much about that. This is more typical of what you get on a newer and an older Illumina machine, sorry. And one of the things to notice here, which is very, very typical of Illumina sequencing, is that, sorry, at this end, the quality is a little bit low. And then it goes up and it peaks in the middle of the read, and then it drops down again. And this is very, very typical of Illumina sequencing, and it tends to be worse on the read 2 than on the read 1. In this case, it's fine. So Fastqc have also very helpfully color coded their plots. So they've put red in these bases are really bad region, yellow in these bases are you probably want to be a bit concerned about them region, and anything that's in the green region is probably fine. So although it does go up and down, with this particular data set, I would be quite happy to just use this as it was. Sometimes what you see happens with these is it starts here, it goes up, and then it goes right down like this. And this is when you need to be thinking about trimming your data. So if you remember, Omar was talking about trimming tools, and what trimming tools do is they will take each read individually and they'll trim off bad bases from the 3' end, working inwards until they get to a region where the quality is good. But for this particular one, this is an example of a reasonable data set I'd be quite happy to use. While Fastqc is reading your Fastq file, it takes the opportunity to give you a load of other data as well. So I'm not going to go through every single one of these, but I will touch on some of the ones that are more interesting, I think. So can we go to the per base sequence content? Okay, so this is, again, we have the position in the read from left to right on the x-axis. And on the y-axis, what we have is the percentage of reads, sorry, percentage of bases at that position that are each of the different nucleotides. So the red line is showing us the percentage of bases that were T, and the blue line, the percentage of bases that were C, and so on and so forth. One of the things you'll notice with this is the big red X up here. The reason for that is twofold. So Fastqc actually assumes that you're doing DNA sequencing, but in this case we're not. We're doing RNA sequencing, and this data set is stranded. So what Fastqc assumes is that the percentage of T will always be equal to the percentage of A, because with whole genome sequencing, you're sequencing both strands, and so you'd expect for every T there would also be an A. And likewise, the percentage of G should be the same as the percentage of C. And the reason this is telling us that this is bad is because you can see here the percentage of T is consistently higher than the percentage of A's, and the percentage of G's is consistently higher than the percentage of C's. And the reason that happens is because this is a stranded library prep, so we're predominantly sequencing the coding strand, and there's a coding strand bias. So although in the DNA for every C there's a G, in the mRNA, because it's single-stranded, that's not always the case. And so in this case, this is an example of where Fastqc is telling you that your data is bad, but you have to understand what your data actually is and what the underlying assumptions of Fastqc are. And in this case, this data is actually fine. There's nothing wrong with it. It's just that it's a stranded RNA sequencing experiment, and that's not what Fastqc is expecting. Another good one to look at is, yes, this one, the percentage, the sequence GC content. So again, we have, oh no, we don't actually this time. We've got mean GC content on the x-axis, and then this is basically a histogram. It's a frequency on the y-axis. Fastqc takes the median GC content from your data and uses that to construct a theoretical distribution of GC content that's shown in blue. And then what it actually observed is what's shown in red. And this is the reason this one's failed is because it's jagged and it's spiky and it's all over the place. And again, this is a case of understanding what your data is and what your organism is and what Fastqc is expecting. So again, Fastqc is expecting you to sequence the entire genome. So it's expecting you to sequence intergenic regions as well as exons and coding sequences. But of course, if you're doing RNA-Seq, you're only sequencing the coding sequences. And that can give you some bias. So for example, in some or many organisms, actually the coding sequences are much more GC rich than the genome is, than the intergenic regions are. And that can lead to plots with weird peaks and stuff like this on them. The other thing that's particularly causing an error on this one is the fact that it's slightly bimodal. So if we were to smooth this red peak out, it would sort of go like that, have this shoulder on it here. And this can be several things. It can be an indication that there's contamination. So that's something to keep an eye out for. So often if you have bacteria in your culture, the bacterial GC content will be different from the genome, the organism you're actually sequencing. In some parasites which have big complicated organella genomes, this can actually come from the organelle. So it can come from, for example, I often see this in Leishmania where you get kinetoplastid DNA causing this kind of effect. But the other thing this could be is if you have, I think this was probably an insect data set, is that right? So if you've got, for example, gut from your insect in your sample, and that has bacteria or some other parasites or something in it, that could cause this kind of thing as well. So it's something to keep an eye on. I'd be much more worried about this peak if this was a culture, because I might be thinking, oh, I've got bacteria in my culture which I shouldn't have. But actually from a whole insect, I would expect this to look kind of a bit weird and spiky like this anyway. So again, this is a case of understanding your data and what the assumptions that Fastqc are making actually are. The very last thing to look at, I think, is probably the overrepresented sequences. And this lists sequences that we see in the sample you've sequenced more often than you would expect to see by chance. And these can arise from several different things. Again, here Fastqc is making the assumption that you are sequencing a whole genome and not the transcriptome. So it's assuming you're sampling everything randomly, but actually you're not, because you're going to sample highly expressed transcripts more frequently. So sometimes these samples come from transcripts that are particularly highly expressed. There's no hit here because this is Fastqc is looking for hits in its database of adapters. So that's the other thing you sometimes see in here is adapter sequences. In this case, we know that this probably isn't an adapter, but sometimes you can find what this is by taking these sequences and blasting them. And what you'll sometimes see with these as well is that it's actually a longer sequence that's been broken up, and you can see where it overlaps. In this case, I would suspect that this is probably something from a very highly expressed transcript in this particular organism. Okay, I don't think I wanted to go on through anything else in the Fastq report, Omar. Should we move on to some of the other data?
Yeah, let me do that quickly.
Yeah.
Let's go back. So maybe, depending on time, Kathryn, and how quickly you can go through stuff, but one thing, let me actually go back to the other data set that had the Fastq output. I believe it was this one here that I looked at.
It was, yeah.
And so DESeq2 has a couple of the main output that you would care about is the actual table that includes your differential expression values. Did you want to talk about the plot quickly at all?
Yeah, I can talk about the table
summary?
quickly. I'll talk about the table quickly first, because that's probably the more important thing for most
Okay.
people. So yeah, I mean, Omar gave a sort of a reasonable description of this earlier, but just to go through this in a bit more detail, this is the output of the statistical test that DESeq2 does. So DESeq2 normalizes, it takes the raw count data, it normalizes them, as we were discussing earlier, and then it does something for every gene. It does something called a Wold test. And a Wold test is similar to a t-test, except it's designed for non-parametric data. So it handles the fact that this data is not normally distributed. So the columns you get back, you get, first of all, the gene ID. So you've got, should have one row for every gene that was captured in your experiment. The next column is the mean, and this is the mean of the expression level, the normalized expression for this gene across all of the samples in your that you considered, regardless of which condition they're in. So if you had 2 conditions with 3 replicates of each, this is the mean of all 6 of those samples. The utility of this really is to exclude very highly expressed genes, which could skew your results, or very poorly expressed genes, and particularly very poorly expressed genes, because if your gene has 1 count in one condition and 2 counts in another condition, it looks like a 2-fold change, but you really have no idea of knowing if that's really a 2-fold change or if it's just noise. So we quite often want to filter those out. So this column is quite often used for ranking and filtering. The log 2-fold change is basically the differential expression value, and we use a log 2 because when we put the this into log 2, it means that if the genes aren't changing at all, the value will be 0. Positive values means that your gene is upregulated, and negative values means that your gene is downregulated. And then the magnitude of the number gives you an idea of how far it's upregulated or downregulated. So yeah, you can see there's a lot here where you know, the magnitude is very small, but there's some here where we've got good upregulation. You have the standard error of the log 2-fold of the fold change across your replicates, and then we have the actual statistical test. So as I said, the statistical test that DESeq does is something called a Wold test. And just like when you do a t-test, I think most people here will have at some point done a t-test manually. And when you do that, you calculate a value for t, and then you look it up to get your p-value. And that's exactly how a Wold test works as well. So the Wold stat is the equivalent of your value for t in a t-test. And then DESeq2 does the work of looking that up for you. So it gives you then the p-value in the next column, and then the adjusted p-value that Omar was talking about earlier. And as Omar said, it's really important to use the adjusted p-value for ranking or for setting cutoffs or whatever you want to do with your data.
The second, if you look at the second row, that's a pretty big difference, right, between the p-value and the adjusted p-value. And so there will be situations where it actually makes a big difference. In many cases, it doesn't. Like they're both, you would potentially consider both of these statistically robust, but in other cases, it actually makes a big difference.
Yeah, so there's a question in the chat which says p adjusted is the Q value. So yeah, absolutely, yes, that's correct. So sometimes you'll hear it called the adjusted p value, sometimes you'll hear it called the Q value, and sometimes you might hear it called the FDR. But they're all basically the same thing, and that's the one you should use.
Cool. So see. Maybe we can quickly look at this PDF output then of the seq2.
Okay. This is a good example of one that doesn't look good, but there you go.
Right. Exactly. Yeah.
So the other thing that DSeq2 gives you is, excuse me, some plots that can help to help you with quality control of your data. So we already looked at the quality control of the actual sequencing, but this is more about looking at, if the sequencing was good, it's more about looking at, are the replicates what you expect them to be, and what's the variance in your data like, and those kinds of more experimental aspects, rather than just did the sequencing work or not. So I may be a bit surprised with this, because DSeq2 has actually changed that. Galaxy's changed what DSeq2 is giving you recently, so some of these aren't going to be what I'm expecting them to be. But the first thing you can see here is a Pca plot. And so the Pca plot plots the first two principal components of your data set. So these are the two aspects of your data set that contribute the most variance to your experiment. And then they plot those points on those principal components. And for single factor experiments like this, the best utility of this is to check how your replicates look with respect to each other. And this is actually an example of one that looks bad. So what you can see here is on the x-axis, the x-axis accounts for 78% of the variance in your experiment. And what you can see here is we have this the two pink samples. These are replicates of each other, so these are both the mutant. And there's a massive gap between them on this x-axis. So this is telling me that most of the variance, nearly 80% of the variance in my experiment, is caused by a difference between two replicates that should be very similar to each other. The wild type looks better. So although there's some variance in this axis, this is only contributing 17% of the variance in our experiment. But if we look at how far apart they are in this axis, they're actually very close to each other. So this is good. I'm okay with the wild type here. But there's a problem with the mutants. And to me, this means that actually I wouldn't be happy doing the differential expression for this, because there's so much gap between these mutants, you're just going to see noise. You're not really going to see anything that's worthwhile. So there's a question here. In a triplicate data set, if we find that one replicate does not look like the other two, would it be better to rerun the analysis without the outlier experiment? It depends. You can do that, but it also depends if you can see where that variance is coming from. So in this case, the only information we have is mutant and wild type. And in this case, if I were doing this analysis for a lab, I would actually just say redo your experiment and maybe add another replicate next time. If you've got lots of replicates, you can maybe take some away, but it's not the ideal thing to do. But sometimes you can find something else that accounts for this. So maybe you had a postdoc who did mutant 1 and wild type 1, and there was a student shadowing them who did mutant 2 and wild type 2. And in that case, you may be able to model where that variance is coming from in your analysis. And that's a better solution if it's possible for you to do that. In most cases, if the lab has the money, though, I would advise them just to restart the experiment, to be perfectly honest. In cases where the quality of RNA is low because of conditions used in the experiments, we've seen higher variance. So yeah, and of course you can get very high variance, and you can get very high variance from things like field isolates and so on as well. My problem here isn't necessarily the magnitude of this variance, but it's the fact that the two mutant samples, there's much more variance between them than there is between this mutant sample and the wild type samples. So with only two replicates, if you're doing a differential expression, what it's going to show you is what's happening between here and here, because this one is going to create all the noise basically. But because there's both conditions over on this side and you've only got 2 replicates, you still can't really tell where the signal is coming from. But yeah, absolutely. If you've got lots of variants, that's fine, but you still need your replicates to be closer to each other than they are to other samples. So this is another way of visualizing the same thing. This is done using Euclidean distance rather than variance. And again, this is demonstrating that this one wasn't a particularly good experiment. Ideally, what you'd see here is the more similar samples are, the darker the blue, and the light ones indicate samples that are very dissimilar to each other. And again, here you can see that mutant 2 is very dissimilar to mutant 1. And mutant 2 is, as you'd hope here, most similar to itself because they should be identical. So that's basically another way of visualizing the same thing using a slightly different metric. This is to do with how DSeq2 transforms your data. So RNA sequencing has a property which is it's known as heteroscedasticity, and what that means is that the variance is not equal over the range of the data. So if you look, the variance at this end is very low, and the variance at this end is much higher. So here, the raw data, that the black points are the normalized data before it's been transformed, but DESeq does a transformation to try and make it fit, make the variance more even, and that's what the blue points are showing. And so you can get a sense from this for how successful that transformation was, and in this case, reasonably successful. And the blue points up here show individual data points that couldn't be transformed. It's okay to have a few of these. These are actually normally your highly differentially expressed genes. So it's okay to have a few like this, but if you've got lots of them, or if your data is still looking very heteroscedastic after the transformation, then again, you need to revisit what you've done and how good the sequencing was. And then here we've got a histogram of the p-values for this comparison. And you'd expect here that the majority of your genes are not changing. So you'd expect the majority of your genes to have p-values in this range, and that's fine. That's exactly what we see. And a smaller number are changing. And that's fine. That's exactly what we see in this case. Something's weird with this MA plot. The MA plot we've plotted, this is another way of looking at heteroscedasticity. We've got the mean on this axis, so this is showing us the range of our data, and then we've got the log 2-fold change on the y-axis. So genes that are up here and genes that are down here are going to be the ones that are significantly different from each other. And often, depending on how this plot's been made, you'll sometimes see that those are colored red or colored blue or colored another color. What we're looking for here is that the data, the ones that are upregulated, are sort of evenly arranged along this axis. It's not like you have nothing upregulated here and then everything at this end is upregulated. So we don't want MA plots that are this shape if we can avoid it. Again, this one actually looks okay.
That's it in terms of what DESeq2.
That was it, yeah. Of course, if you're using DESeq2 at the command line in R, you can generate all sorts of other plots from it as well, but these are the ones that Galaxy gives you by default.
Cool, thank you. Are there any questions for Kathryn? I'm just going to quickly switch to this male versus female. I'm curious to see what it looks like. These are just samples that came from the sequence read archive. So obviously there's no knowledge about, sometimes there are mistakes in what samples were uploaded there and so forth. So that's something to keep in mind. But I was just curious if that's so yeah,
this
here's a better
this
one.
This one's a PCA plot that looks good. So we can see here that all the males are over here, all the females are over here. That's accounting for more than 90% of the variance. It is just the difference between males and females. And if that's the contrast you're trying to do in your differential expression, then you're going to get really good signal for that, and it's not going to be messed up by noise from other factors.
Cool. I'm glad there was one that looked good.
Yeah. It's good to show what bad ones look like as well.
Oh, sure, absolutely. And I guess it's a nice warning, maybe, or I guess asterisk that there is all this data in the sequence read archive, and sometimes they're either mislabeled there, or something is is wrong in the experiment, and the data is available there. So being able to look at these plots, even if you want to reanalyze somebody else's data, looking at these plots and confirming that, well, what they said they discovered is reflected in the data that's deposited, is quite useful, because sometimes it doesn't mean their observations were wrong. It just means maybe the samples that were loaded were wrongly labeled, or potentially they just uploaded the wrong file and so forth. So there's some questions coming up in the thing. Let's see. So this plot,
I have
for
to
Nadia,
go very quickly, but I can just answer these quickly. So someone asked about the PCA plot, and can you adjust the percentage of the variant on either axis? And the answer to that is no. So what this is doing, it's doing a PCA analysis, and if you don't know what that is, there's some really good resources for this on StatQuest to look at, which can explain it much more clearly than I can. But essentially, the 94% here is the variance that was observed in your experiment. So the only way to change that is by doing your experiment again, or doing your experiment in a slightly different way. You can't just change it in the plotting. That's just what it was. And there was a question about this one. This one doesn't really have a name. I just often call it a sample distances plot. Personally, I find it less useful, so I don't often make this one. I'll normally just make the PCA plot. The Euclidean distances can be a bit misleading sometimes. And there was a question about sorting unmapped reads and duplicates. So do you mean when you're counting the file, Mohammed, or do you mean when you're doing the alignment?
So we'll see if Mohammed answers, but I think.
Yeah.
Can you hear me?
Yes.
Yes.
Yeah, for alignment.
For the alignment. So your alignment will be in a format called BAM, and there's a set of tools called SAM tools, and that gives you a whole lot of different options for filtering, and you can use SAM tools to find unmapped reads and to also find duplicate reads as well. So yeah, that's what you want to be looking for, is a tool called SAM tools.
And then the last question is what parameters we should take care while comparing data across different RNA sequencing studies.
So the biggest thing if you're comparing across studies is to do a PCA plot and get as much metadata about those studies as you can, because if you've got RNA-Seq that's come from different studies, you will have batch effects between those different studies. And so it's important to understand what those batch effects are, what the magnitude of those batch effects are, which will give you an idea of whether it's even possible to do the comparison you want to do or not, and whether there's any way of regressing it out. So for example, if you're doing a drug resistance study and you've got wild type and drug resistance in study 1 and wild type and drug resistance in study 2, you can still do that comparison even if there's a batch effect, because you have both conditions in both studies. But if you have one study with only the wild type and one study with only the drug resistance, then you have no way of knowing if the effect you're seeing is a drug resistance effect or just a batch effect. So it's really about understanding the sources of variance in your data, how big they are, and how balanced the studies are relative to each other, which will enable you to get rid of those unwanted batch effects.
Cool. Well, I know you have to run, Kathryn, so we'll let you go. Thank you very much. Again, Kathryn is an expert with this kind of data. She actually does the work of analyzing many of these types of data sets. And if you have additional questions about this, feel free to drop us an email. You can ask us, but we'll probably not do a fair job answering the question. We'll try, but if you drop us an email, we can always get Kathryn's input on those responses. So if you have a specific experimental question you have for your data, definitely drop us a line and we'll try and help. Okay, so let's see, where are we? Okay, so we're obviously running late. So what I'm going to do is, let's see, what should we do? So let me go ahead and quickly talk about ontologies. I'll try and do this again as a quick lecture, and then maybe we can do the Go enrichment exercise, and then we can decide about the phenotypic data. Maybe we can postpone the phenotypic data discussion until after the break. The phenotypic data will be very quick. It's just a description of what kind of data we have in the resource. And so I'll move this up to right before the actual, right after the break. So we'll do that to make it a bit easier on everybody. Okay, so there you go here. Okay, so okay, so we're going to talk a little bit about Go terms, EC numbers, and enrichment analyses. And there's often a question about, well, what are Go terms and why are they useful? EC numbers, also you can apply the same thing to it. And so for the purpose of our discussion, Go terms stand for gene ontology. And so what is an ontology? Let's start with that. And the ontology really is just a formal description of a particular field. And also it provides information about the relatedness or relationships between the different descriptors in your whatever field you're interested in. And so, for example, we can generate a formal description of the field of science, which we can break it up. There are many ways of breaking it up, but let's say traditionally we can break it up into three different arms: formal sciences, natural sciences, and social sciences. And then each one of these can be broken up into multiple other subcategories, like formal sciences can include logic and mathematics. Natural sciences can include life and biological sciences, where most of us are located, and physical sciences. And then social sciences would be something like sociology. But the key here is that you are providing a formal description for each of these aspects of science, and you're showing the relationships between these. There may be some relationships that transcend these different arms, and also all of these can have multiple children or parents that give them a hierarchical relationship in this particular ontology. So here, this is basically, we've developed an ontology for science, and it's a very simple one, a very basic one, and we're showing the connectedness between them. And so what the gene ontology tries to do, it's really a way to describe all the knowledge we have of the field of biological sciences. Okay, and basically the gene ontology is broken up into three broad categories. Those are molecular function, cellular component, and biological process. Each one of these obviously will be broken up into many hundreds or thousands of subcategories and with lots of interconnectedness and relationships. And so what do we mean by these different branches of the gene ontology? The molecular function refers to the activity of at the molecular level. So for example, a toxin would be or toxin activity, for example, or transporter activity, like shown here in this image, a protein or a gene that encodes a protein that is a transporter can have a molecular function of a transporter activity, for example. Okay, and so the idea here is that you can take every gene and if you can formally describe the function of every gene, you can put them into these different categories. Then you have a standardized way of comparing things across your genome and also across different genomes. So cellular component will tell you something about where the function of a protein takes place. So for example, it could be in the flagellum or cilium, or it could be in the mitochondria, for example, right here. So a protein can be exerting its function in the mitochondria, so it'll get a cellular component Go term of mitochondrion, and there could be obviously subcategories. And then finally, the biological process tells you about the actual process that the protein is functioning in, like a biochemical pathway. So for example, pyrimidine biosynthesis or the pentose phosphate pathway would be part of the Go term, would be part of this arm of Go. Okay, so that's sort of the basic idea. So this becomes very useful when you're trying to compare things within a genome or across genomes. Because as we know, people call things different things, right? So I can, if I annotated a gene that I'm working on in the lab, and I notice that the gene, causes my bacteria to turn red, I may, if I knock it out, I may call it red-inducing, protein 1. Somebody else in a different lab may realize that that same protein is upregulated when you feed the bacteria a particular type of sugar, right? And so they may call it a sugar protein 1 or sugar transporter 1, and somebody else may do something else. And now the same gene, right, described in the same bacteria may have different names. And just looking at the names, you will not know that these are connected. So red gene 1 and sugar gene 1, just by the name, you will have no idea that they're connected. But if you formally describe them and use an ontology to describe them, you may end up looking at it and saying, well, red gene 1 and sugar gene 1 are actually both a transporter, and maybe the activity is transporting sugar, right? So now all of a sudden there's a connection, an ontology that connects these the gene. First of all, the description of the gene, which is described differently by different groups, and also, of course, across organisms. So the same, red gene 1 in bacteria 1 and red gene 1 in humans, right, may have totally different names, but they both may have the same transporter function. And knowing that is quite useful, allowing you to find connections between across sort of organisms, but also within your organism. And an example of this is looking at how people describe these different organelles in different organisms. So this is called a cyanide in these free living photosynthetic organisms. It's called a chloroplast in plants, and it's called an apicoplast in parasites like Toxoplasma or Plasmodium. They have different functions, but they all have the same evolutionary background, and as a result, even though this one is called a cyanide, this one is called a chloroplast, and this one is called an apicoplast, they all get the same Go term, which is plastid. They may have subcategories in here where there's a subcategory for apicoplast or a subcategory for chloroplast, depending on function and the organisms they belong to and so forth. But ultimately, you can now connect these together by virtue of that they have the same Go term, which is that they're all plastids. Okay, is that, hopefully that's clear so far? I think it's a very basic concept, but often people are not sure exactly why that's potentially useful. And so now if you have a set of results, actually before I talk about enrichment, let me talk quickly about EC numbers. This again is just another way of formally and very methodically describing enzymes. And so the nomenclature is called the enzyme commission number, and it's composed of 4 digits. Each digit signifies something. Each place signifies something. So the first digit which is, and they're typically, they can be 1 or 2 digit numbers. Those typically tell you which class the enzyme belongs to. And this is, again, this is a formal description of enzymes specifically. The second, so you have the class that an enzyme belongs to. So for example, hydrolase. The second one is a subclass. So this could be a hydrolase that's acting on ester bonds. And so the number 3 would indicate hydrolase, the number 1 would indicate that it acts on ester bonds. The third figure is again another subclass of this, and so it tells you that it's a phosphoric. And then finally, the fourth one tells you again it's additional. I know each of these is a subclass of each other, but basically having all 4 of them, if you have an enzyme with 4 digits, it means you know exactly what it does down to its actual activity. And so in this case, the 37 would tell you that it's acetoheptulose bisphosphatase. And so so if you have an enzyme that's characterized with 4 digits, then you know exactly what it does. And this helps you then place enzymes or place genes into metabolic pathways, right? Because now you know exactly what this enzyme is. And so if this is assigned to a gene, then that gene, that product of that gene, can be the enzyme that's in the pathway where acetoheptulose 1,7-bisphosphatase is working. And so you can take these EC numbers, plot them on metabolic pathways. And this is what we do in VEuPathDB. So we have metabolic pathways in VEuPathDB, which, and the way we populate these pathways, you'll see here, all of these boxes are EC numbers. And in fact, maybe, well, I won't show it right now, just sort of insert time, but we can, if we have time at some point, we can show you the metabolic pathways in VEuPathDB, but you can say, well, show me glycolysis for in FungiDB, and then you get it, and you can filter this based on particular organism. Sorry, my dog is acting up here. And so you can then see where enzymes are. So you can link enzymes in a pathway to the genes they belong to. So having a formal way to describe enzymes or genes based on function allows you then to start asking for enrichment in your results. So if you have a set of 500 genes that came out of a transcriptomic experiment that are highly upregulated in your specific condition, then you'd like to ask, well, is there something enriched in my sample? And if you're only going to rely on the names of the genes, that sometimes ends up being not terribly useful, because if I called a gene a sugar transporter, if I call it a red producing gene, and somebody else called another sugar transporter a glucose gene 1, then there's no way for me to be able to computationally connect these with each other. I just won't know that these are actually both. Hey, sorry about that. There's no way for me to then connect them to each other and make some inferences based on that connection, right? Okay, so Go terms, where do we get the Go terms from? So Go terms typically are either assigned by an annotator, or we transfer them automatically using computational tools. And the most frequent method we use for automatic transfer is by a tool called Interpro Scan to Go. So Interpro Scan is a tool that will look for any kind of functional domains in your genes. And these functional domains are Interpro is a resource that includes multiple different tools for defining functional domains. One of them is PFAM, you may have heard of, or ProCite, or smart domains. Those can be potentially algorithmic domains, which are looking for patterns that are, it finds sequences that occur, across different organisms, or it could be, real functional domains based on experimentation. So, for example, a kinase domain may have a particular sequence that it will look for, and then you can say, well, this gene, I don't know what it does. Even if it's called red protein 1, it has a kinase domain in it. So now you'll assign, if it has a kinase domain, then you can use the Interpro Scan to Go to assign a Go term that's somehow related to kinase function. Or if there's a transporter domain, let's assume there's one, then you can assign a Go term that gives it the transporter domain activity. So in the case of the red protein and the sugar protein, which are both maybe belong to the same family of transporters, if these two proteins are similar to each other or have a similar domain in them, then you can connect them to each other by virtue of assigning a Go term to them. Or if an annotator assigned the same Go term to them, then they're connected. So now when you do an enrichment analysis on using Go terms, you have a consistent vocabulary that you're using to query in your results. So you're more likely to find things that are meaningful. And again, the same thing applies here with Go terms. You're doing multiple tests over thousands of Go terms in a genome. And so the sample size now starts affecting your p-value or the robustness of your p-values. And as a result, you'll end up correcting for that and doing a false discovery rate, usually a Benjamini-Hochberg false discovery analysis. And we provide those values as well. And so again, as recommended with RNA-Seq to look at the Q value, you definitely want to look at the adjusted p-value, not only the p-value when you're looking at Go term results. And then this is just a quick output. You will do this today in the exercise, but you typically, when you do a Go enrichment, you will get something like this. So out of your gene results, it'll tell you that, let's say this Go term here, which is a serine type endopeptidase activity, is found in 363 genes in your background, which is the genome. Out of those, 18 were in your results, right? And then there's each of these is described. I won't go through all of them, but you can see the fold enrichment in your result and also a p-value associated with this. So you can decide whether you are confident that this is happening. It's not a random event, basically, in your Go enrichment analysis. Okay, and I think this was the last slide. Yeah. So quickly see if there are any questions. Don't think they are. So what we will do today, or for the first exercise, we're going to break out into the breakout rooms. And again, the whole idea, you'll find another description here. Should have a little graphic here. For some reason, it's not loading on my window, but this gives you sort of a description of what I just went through. But then it's going to walk you through an example of running a search and then doing a Go enrichment, so you can take a look at the results. And what I would recommend when you do the enrichment analysis, you will notice that there will be multiple options for different enrichment analyses. There'll be a Go enrichment analysis. There's a metabolic pathway enrichment, which is based on EC numbers. So EC numbers, because it's a consistent way of labeling enzymes, you can use that to do enrichment. And then we also have something called a word enrichment. And so we also recognize that there are many genes that don't have Go terms that are specific to a particular organism. So for example, you can imagine having an expanded family of whatever variable proteins that don't have an official Go term in the Go gene ontology, but they're all called consistently variable genes, and so being able to see whether the word variable is enriched in your results is also useful. So we have but you can do it statistically using the word enrichment. One cautionary word is that obviously if your genome does not have many Go terms assigned to it, then of course your Go enrichment analysis is not going to be robust. And of course, if your genome was poorly annotated with Go terms or had a poor Go term, let's say there was, I don't know, a rogue annotator went in and just started randomly assigning Go terms everywhere, right? That's going to cause problems in your analysis. So it's only as good as the Go terms. Obviously, that's typically not a problem, but it's always good to take a look. Like if you're getting some interesting enrichment, then go take a look at the genes that are in your enrichment. So for example, these 18, I can click on that and go and look at those 18 genes and say, well, do they make sense? Are these all, do they all look like serine endopeptidases? Do they have the right domains in them and so forth? And you can do that in the database. So always check the results. Okay, so are there any questions so far? Okay, so then what I would suggest, so it's already almost 11 o'clock, and we were going to go until 11. So what do people feel like doing? So we can go into breakout rooms right now and work for about half an hour and then take a break. Should we do that? I think we should do that. That's my opinion. Can I have thumbs up or thumbs down? If people think that they need a break right now, we can do that. Okay, I'm seeing a bunch of thumbs up. Great. Okay, perfect. So Rachel, we can go into our breakout rooms, and then maybe in about 30 minutes or so, we can take our break and then come back to do some phenotypic analysis.